was based on DNA homology of at least 70%. While this served as a foundation, it often lacked the resolution to capture the true diversity within the bacterial species. So with the advancements in genetic analysis, particularly with whole genome sequencing, this has revolutionized the field of microbiology. Researchers now use metrics like whole genome average nucleotide identity or ANI to compare and delineate bacterial species with much greater precision. So this has led to a re-evaluation of established taxonomic relationships. I'll take a minute to give an example here. Looking back at India's first wave of SARS-CoV-2 pandemic, where various variants dominated different regions with a notable shift in variants by April 2020. The wave peaked in September 2020 and began to decline thereafter. However, the country noticed a resurgence of cases in January 2021, predominantly in 3 states, that is Maharashtra, Punjab and Chhattisgarh. In Maharashtra, apart from the major cities like Mumbai and Pune, a significant rise in cases was seen in districts of Vidarbha, in particular Amravati. The emergence of new global variants around the same period was concerning as they were suspected to be responsible for fresh surge of new cases. On sequencing these samples from Amravati, it was found that Amravati was dominated by clade 21b, which carried immune escape mutations. Due to the presence of these immune escape mutations, the WHO on 4th April 2021 declared Kappa variant as a variant of interest. This variant rapidly evolved to what is now known as Delta variant and was responsible for driving India's second wave. Therefore, when a continuous surveillance system is implemented, it ensures timely and effective public health responses to such emerging variants. It is not just about identifying the variants, the key lies in establishing their clinical relevance. By correlating genetic findings with real-world clinical outcomes, we generate actionable data that can guide targeted public health interventions. This integrated approach is crucial for shaping effective policies, ensuring resource allocation that aligns with emerging threats, and ultimately improving population health outcomes. Pandemics are exacerbated by urbanization, climate change, and zoonotic spillovers. The recent surveillance initiatives by various organizations worldwide now prioritize high-risk viral families. Now, having discussed the global challenges and initiatives to tackle pandemic risks, the next crucial question is how can we ensure timely responses to these threats? This brings us to the importance of early detection systems. Early detection system is critical in preventing and controlling outbreaks, particularly in low and middle income countries that face a higher risk of zoonotic spillover due to limited resources for clinical surveillance. One promising approach here is what is known as wastewater surveillance. Historically, this tool has been highly effective in monitoring poliovirus and responding to outbreaks. And more recently, it has demonstrated its utility with SARS-CoV-2, influenza, RSV, measles, monkeypox, and Zika. When rates of clinical testing are low, wastewater surveillance provides a more accurate picture of the actual burden of the disease in a community. By detecting pathogens shed into the environment, it bypasses the need for individual testing, identifying both symptomatic and asymptomatic cases. This capability makes wastewater surveillance particularly valuable in under-resourced settings or during large-scale outbreaks. It can detect pathogens irrespective of their transmission route and correlate findings with disease incidence in the population. Now, by integrating such strategies into our existing surveillance frameworks, we can bridge gaps in traditional monitoring systems and significantly enhance our ability to respond to emerging health threats. Now here are some real-time examples from Pune, where SARS-CoV-2 variants like JN.1 and KP.2 were detected in wastewater 3 months and 1 month respectively before the rise of clinical cases in the community. So these findings highlight the effectiveness of wastewater surveillance as a reliable early warning system for outbreak detection and response. Now the next logical question is, how can we expand such approaches to prepare for an inevitable next pandemic? We live in a world where emerging health threats dominate the headlines, be it Mpox virus, rising cases of bird flu, or fears of Marburg virus outbreaks. The critical question is not if the next pandemic will happen, but when. So how do we prepare for it? One promising solution is investing in wastewater surveillance, which offers numerous benefits, that is preparedness, scalability and cost effectiveness, and supports data-driven public health measures. So as we brace for future health crisis, incorporating wastewater surveillance into our public health frameworks is not just an option, it's a necessity to stay ahead of emerging threats. The next is antimicrobial resistance, as we call it as the silent pandemic. It poses a significant threat to global health. Misuse of antimicrobials continue to drive the evolution of resistance, calling for innovative solutions, improved surveillance, and public health education. Genomics is at the forefront of addressing this crisis through several key applications, either by augmenting the phenotypic testing that we do or understanding the gene diversity for better characterization of these genes and their variations. It helps in detection of common antibiotic resistance genes and at the same time addresses the challenges of undetected genes, thereby filling the gap by uncovering the hidden resistance mechanisms. The studying AMR evolution in pathogens is also important. Cases once thought to involve new strains often reveal evolutionary adaptations of existing strains, reshaping our understanding of infection dynamics. AMR is not just a human health issue, but a One Health challenge affecting humans, animals, and the environment. Genomics helps to track the transmission of AMR genes between humans, animals, and environments, enabling targeted interventions. By integrating genomic approaches into One Health strategy, we can better understand the interconnected drivers of AMR and develop comprehensive solutions to address this multifaceted challenge. Now let us shift our focus to the healthcare settings. Healthcare-associated infections remain a persistent challenge in healthcare settings despite the implementation of infection control strategies and bundles. Now, traditional microbiological methods often fall short when it comes to determining the true relatedness of organisms, which is crucial for understanding the spread of infections. Now, this is where NGS plays a transformative role. It allows us to pinpoint the exact source of an outbreak. It provides detailed insights into how pathogens are transmitted between patients, between healthcare workers, and even between healthcare facilities. NGS offers a comprehensive view of how antimicrobial resistance genes evolve and spread within and across the healthcare settings. Now, I'd like to give a very interesting example here. Building on the role of genomics in infection control, we conducted a study at BJ Government Medical College in collaboration with Johns Hopkins University. In this study, what we did is we sequenced 11 E. coli and 15 Klebsiella pneumoniae carbapenemase resistant strains, and we wanted to see what were the resistant genes present. As expected, we detected the presence of carbapenemase resistant genes, various carbapenemase resistant genes. However, what stood out were additional findings beyond antibiotic resistance, and we also detected what is called today as disinfectant resistant genes in these isolates. So this discovery has significant implications for each and every infection control practitioner, as these genes can potentially reduce the efficacy of routine disinfection protocols in healthcare settings. The detection of disinfectant genes in carbapenems resistant isolates not only highlights a new dimension of antimicrobial resistance, but also raises a critical implication for hospital infection control. This can lead to persistent contamination of hospital surfaces and equipment, may increase the risk of healthcare-associated infections, particularly in high-risk areas like intensive care units where patient vulnerability is higher. There can be an increased need for enhanced surveillance of disinfectant-resistant genes, where hospitals should routinely test the efficacy of disinfectants being used to ensure that they remain effective against the evolving pathogens. Additionally, they may also need to explore alternative disinfectants and advanced cleaning strategies to mitigate these risks. Now, another exciting area where NGS has been leveraged is paving the way for innovative diagnostic kit development. It can identify the entire genome of the pathogen and guide the design of region-specific diagnostic tests based on circulating strains. It will also enable the creation of multi-pathogen panels to test for multiple diseases simultaneously, ensuring that the diagnostics stay relevant for rapidly evolving diseases. It will help in generate a genomic database to build